Thank you, everyone, for taking the time to listen to this talk, because it's quite a dry talk. If I can make it interesting for you, I'll try my best. I've got a video, which is always a recipe for disaster, but Gemma has promised me she's going to make it work. Okay, so I want to talk to you about oncolytic viruses, and I'm going to start off by telling you who I am. I'm a head and neck cancer surgeon. I've been interested in oncolytic virus research, immunotherapy, if you like, for the last 2 decades nearly. And about 13 years ago, we decided to take this to the clinic. So translating our research into a treatment for cancer. So what is immuno-oncology, immunotherapy? There are lots of different aspects, and some of it you would have heard of. Everyone now knows about mRNA vaccines. Probably some of you would know about checkpoint inhibitors. These are the latest treatment to gain favor in the immuno-oncology world. And my bit, which is oncolytic viruses. I won't go so much into the other ones, but I want to spend some time talking to you about how this immunotherapy is important, because it completely contradicts everything else in that it's a way to boost the body's natural defenses to attack these cancer cells. So we make cancer cells all the time, millions of them, but the body is continuously undergoing surveillance, and T cells and other cells will recognize these abnormal mutant cells and kill them. But in the case of cancer, there is something that tells the body that I'm actually part of you, so don't attack me. And that's where checkpoint inhibitors have come in. To date, we have over 20 cancer indications approved by the FDA for the treatment, for using immunotherapy as a treatment. How does it work? Checkpoint inhibitors are, if you like, sitting on the surface of the cell, and they tell the body, don't attack me. But if you block these using a checkpoint inhibitor or a PD-1 inhibitor, programmed death ligand inhibitor, then that causes lysis of the cell. The T cells now say, actually, you're not one of us, and I'm going to destroy you because you're abnormal. These are checkpoint inhibitors, and the people who actually came up with this have now won the Nobel Prize. Remarkable work, and is continuing to improve all the time. Well, what about viruses? We've long known that a viral infection can actually have a dramatic effect on cancer. And people who've had viral infections, then their cancers regressed. Remarkable. So people have tried since the 50s and the 60s to use cancers for the treatment, specifically cervical cancer. The first oncolytic virus, oncolytic means that it breaks down cells. The first oncolytic virus was approved in China in 2005, and that was an adenovirus. In 2015, another virus, which is a herpes virus this time, called telamagen laharparepvec, it took me about 4 weeks to learn how to say that, became the first oncolytic virus approved for the treatment of malignant melanoma, stage 3 and stage 4. And then last December, on the 16th of December, the FDA approved the first oncolytic adenovirus for the treatment of BCG-resistant bladder cancer. Really quite remarkable how we've come so far in such a short period of time. Please make the video work.
Oncolytic viruses work by priming the immune system in a manner similar to a vaccine. The treatment involves the injection of oncolytic viruses into an accessible tumor site, primary or metastases. The viruses would then infect tumor cells. They would deliver their DNA into the cell, which then takes over the cellular reproductive mechanism to produce tens of thousands of copies of itself. The virus presents a strong danger signal to the immune system through the release of cytokines and chemokines. Oncolytic viruses would then cause lysis of the tumor cells, releasing tens of thousands of copies of the virus that then infect other tumor cells. When the virus replicates in tumor cells and lyses them, it releases hidden tumor-associated antigens. These new, previously hidden antigens are now recognized by antigen-presenting cells, most commonly dendritic cells. The dendritic cell's main function is to process antigen material and present it on the cell surface to prime naive cytotoxic T cells against these tumor-associated antigens. These newly primed T cells would then go on to attack and kill other tumor cells that have the same tumor-associated antigens not previously recognized by the immune system.
So the virus is effectively acting as a vaccine. In the way that we understand vaccines now, everybody's an expert on vaccines, from mRNA to the AstraZeneca vaccine. So we've got a virus, and it does what it says on the packet, which is it is very good at breaking down cells. In fact, when we compared it to other viruses, we think that we have the most powerful virus in breaking down cells of the same type of viruses. So we then had to just make the virus and produce it in enough quantities for it to go into a clinical trial. Well, of course, that is no easy feat. So what does this virus do? This virus infects the cancer cells, as I said. It starts to replicate within it, and it breaks it down, releasing these antigens, which now become the vaccinating effect, or have the vaccinating effect. Whilst in normal cells, it does not replicate. The replication is inhibited. We've looked at normal cells, and we look at our virus here at the bottom, and it doesn't replicate compared to the wild-type virus and to other controls, including the Onyx 015, which I spoke about earlier on. So this is a safe virus. It does not replicate within normal cells. This work has been published already in Nature Cancer Gene Therapy. But when we try it out on tumor cells, in mice without an immune system, we see a remarkable improvement in survival of these mice with the tumors injected into them that have been then treated with the cancer. We then went on to look at other cancers, every solid tumor that we can get our hands on, and everything we've tested so far, the virus replicates in it quite dramatically. Again, this work is published. More recently, we started to look at sarcoma and renal cell lines, and again, all this shows you is that the virus replicates very rapidly, very effectively in these cancer cell lines. We've made the virus. We have a drug product as of last week. It's going to be what's called fill and finished and labeled and sent across from the US to the UK in August for clinical trials to start in December at the Royal Marsden Hospital under Professor Kevin Harrington, who's the doyen of oncolytic viruses and running clinical trials on them, and Dr. Martin Forster, my oncologist and colleague at UCLH. Our first clinical trial is going to be the first in man, and it's designed such that we use the virus on its own in the first 6 patients to show that this is safe. We're then going to combine it with the old checkpoint inhibitors that I mentioned earlier on, and to make sure that it's safe, and then we're going to go on to further 12 patients to look at efficacy. So safety first, and then we'll look at efficacy. This is the trial design, but I'm not really going to bore you anymore with it. This has been 13 years since we started this work. Next, we hope to go into childhood sarcomas, hopefully if this is all successful, and these are very rare tumors. As a byproduct of this work, we are also conducting a collaborative research project with the Institute of Cancer Research with Professor Alan Melcher to look at why people do not respond to this immunotherapy. Happy to take any questions. Thank you very much indeed.